On Alaska's western edge, where the land stretches toward Russia across the narrow Bering Strait, a cluster of limestone caves has become one of the most important archaeological sites in the search for humanity's earliest journey into the Americas. Known as Trail Creek Caves, this remote location has transformed how researchers think about the first people who reached the New World. Scientific excavations began in the late 1940s under Danish archaeologist Helge Larsen. Working inside the caves on Alaska's Seward Peninsula, his team uncovered an extraordinary collection of evidence: human bone fragments, carefully crafted stone tools, ivory artifacts, and the remains of Ice Age animals, including mammoths, bison, and caribou. Together, these discoveries painted a picture of people surviving in the harsh environment of the late Pleistocene. For decades, however, much of the material remained largely untouched in museum collections. The human remains were stored in Denmark for more than 60 years before being returned to Alaska in 2010 under the Native American Graves Protection and Repatriation Act. Their return gave scientists an opportunity to study them with modern technology that simply did not exist when they were first excavated. Researchers re-examined the bones using radiocarbon dating, anatomical analysis, and, most importantly, advanced ancient DNA sequencing. These new genetic techniques revealed details that earlier generations of archaeologists could never have imagined. Instead of reinforcing long-held assumptions, the evidence suggested a far more complicated history of human migration into the Americas. For much of the 20th century, the dominant explanation was known as the Clovis First Theory. According to this model, the first Americans were a single population of hunters who crossed the Bering Land Bridge from Asia roughly 13,000 years ago before rapidly spreading throughout North and South America. It was a straightforward narrative that appeared to fit the archaeological evidence available at the time and became the standard explanation taught in classrooms for decades. Over the last 15 years, however, discoveries from Alaska, the Yukon, the Pacific Northwest, Mexico, Brazil, and several other regions have steadily challenged that view. Ancient DNA studies now suggest that the settlement of the Americas was not a single migration but a much longer and more complex process. Evidence points toward multiple waves of migration, genetically distinct populations, and even ancient groups that disappeared entirely without leaving direct descendants among living indigenous peoples. The discoveries from Trail Creek Caves fit neatly into this growing body of research. Rather than standing alone, they support an emerging pattern indicating that the earliest chapters of American history were far more diverse than previously believed. Scientists are now investigating not only when people first entered the continent, but also how many different groups made the journey and how they interacted over thousands of years. These findings are gradually reshaping one of archaeology's biggest historical questions. As genetic evidence continues to accumulate, researchers are revising ideas that remained largely unquestioned for generations. What once seemed like a simple migration story is now evolving into a far richer and more detailed account of humanity's expansion into the Western Hemisphere. To understand why Trail Creek Caves has become so significant, it's necessary to look back at the theory it helped challenge. The Clovis First hypothesis wasn't merely one possible explanation. It dominated American archaeology for much of the last century. Its origins trace back to 1932, when archaeologists near Clovis, New Mexico, uncovered distinctive fluted stone spear points alongside the remains of mammoths. That discovery became the foundation of a theory that shaped archaeological thinking for decades, until new evidence began revealing that the real story was far more complex than anyone had imagined. The points were beautifully crafted, large, leaf-shaped, with a characteristic fluted channel running up from the base on both faces. Nothing like them had been found in the Old World. Radiocarbon dating, when it became available in the 1950s, music placed the Clovis culture at approximately 13,000 years ago. For the next half-century, the Clovis points defined the boundary of American prehistory. They represented the earliest unambiguous evidence of human presence in the Americas. The people who made them were assumed to be the first Americans, big game hunters who had crossed from Siberia to Alaska via the Bering Land Bridge when sea levels were low enough to expose the continental shelf, then migrated south through an ice-free corridor between the Cordilleran and Laurentide ice sheets as the glaciers retreated. The model was elegant: a single founding population, a single migration event, a single technological tradition that spread rapidly across two continents. Any claim of pre-Clovis human presence in the Americas was met with intense skepticism. Sites that appeared to predate Clovis were challenged on methodological grounds. Researchers who advocated for earlier dates found their careers marginalized. The Clovis first consensus was enforced not just through scientific argument, but through institutional pressure. And then the evidence began to accumulate. Monteverde in southern Chile produced evidence of human occupation dating to approximately 14,500 years ago, over 1,000 years before Clovis. The site preserved wooden structures, stone tools, and plant remains in a peat bog that had protected them from decay. The Monteverde dates were challenged. They were defended. Additional excavations confirmed them. Meadowcroft Rock Shelter in Pennsylvania produced dates extending back potentially 16,000 years or more. The Buttermilk Creek complex at the Friedkin site in Texas yielded stone tools beneath Clovis deposits with dates approaching 15,500 years. Each site faced intense scrutiny. Each site survived that scrutiny. The pre-Clovis evidence was real. The decisive break came not from archaeology. It came from genetics. Ancient DNA changed everything. The technology that made human ancient DNA recovery possible was refined primarily in Europe, where cold temperatures and favorable burial conditions preserved genetic material in skeletal remains dating back tens of thousands of years. Applying those techniques to the Americas was more difficult. Warmer climates, different burial practices, and the acidic soils common in many New World environments degraded DNA more rapidly. But the techniques improved, the sample requirements dropped, music, and by the mid-2010s, researchers were successfully extracting and sequencing genomes from skeletal remains scattered across the Western Hemisphere. The first results confirmed long-held ideas and then complicated them. The Anzac child, an infant burial from Montana associated with Clovis artifacts, was sequenced in 2014. The genome showed that Clovis people were ancestral to modern Native Americans and that they represented a population already diversified from the original founding lineage. The Kennewick Man, a 9,000-year-old skeleton from Washington state that had generated years of legal controversy over whether he was Native American, was sequenced in 2015. The genetic analysis was unambiguous. He was closely related to modern Native American populations of the Pacific Northwest. The Spirit Cave mummy from Nevada, approximately 10,600 years old, was sequenced in 2018 and confirmed as Native American, leading to repatriation to the Fallon Paiute Shoshone tribe. These early results established the methodology and confirmed the basic framework. The people who inhabited the Americas at the end of the Pleistocene were ancestral to the indigenous peoples who lived there when Europeans arrived. But they also revealed something unexpected, something about population structure. The ancient Americans were not a single homogeneous population that spread uniformly, music, across two continents. They were multiple populations, genetically distinct groups that had diverged from each other thousands of years before the Clovis horizon, that had taken different migration routes, that had occupied different regions, and that had music left different genetic signatures in their descendants. The single migration model was breaking. The decisive finding came from central Alaska. In 2013, archaeologist Ben Potter of the University of Alaska Fairbanks was excavating a site called Upward Sun River, located in the Tanana River Valley of interior Music, Alaska. The site dated to approximately 11,500 years ago, post-Clovis, but still among the oldest human occupations in Alaska. Potter's team discovered something extraordinary: music. In a residential pit feature beneath a layer of burned material, they found the remains of 2 infants. The preservation was exceptional, and the burial context was undisturbed. The remains were subjected to ancient DNA analysis by a team led by Eske Willerslev of the University of Copenhagen. The results, published in Nature in 2018, rewrote the peopling of the Americas. One of the Upward Sun River infants, designated USR1, yielded a complete genome. That genome did not match any previously known Native American population. It represented a distinct lineage that had diverged from the ancestors of all other Native Americans approximately 20,000 years ago, and that had remained genetically isolated in Beringia for thousands of years afterward. The researchers called this population the Music Ancient Beringians. The ancient Beringians were not the ancestors of most modern Native American groups. They were a parallel lineage, a population that had split from the broader Native American founding population while both groups were still in Beringia before the migration south began. The implication was profound. The peopling of the Americas was not a single wave. It was at minimum 2 waves: the ancient Beringians who remained in Alaska and the ancestral Native Americans who continued south to populate the rest of the hemisphere. And that was before the full complexity emerged. The Trail Creek Caves remains were analyzed as comparative material for the Upward Sun River study and subsequent research. The genetic data from Trail Creek confirmed the Beringian complexity. The individuals buried in those limestone caves on the Seward Peninsula were part of the same deep population structure that the Upward Sun River genome had revealed. They represented the genetic diversity of late Pleistocene Beringia, a diversity that the single migration model had never predicted and could not explain. The Trail Creek data also contributed to the refinement of another model, one that had been proposed on theoretical grounds but was now receiving genetic confirmation: the Beringian standstill hypothesis. The hypothesis, developed by researchers including John Hoffaker of the University of Colorado, proposed that the ancestors of Native Americans had not migrated rapidly from Siberia to the Americas in a single push. Instead, they had paused. For thousands of years, the ancestral population had lived in Beringia itself, the exposed continental shelf that connected Asia and North America during the last glacial maximum when sea levels were hundreds of feet lower than today. Beringia was not just a land bridge. Music. It was a refugium, a habitable region where human populations could survive during the height of the Ice Age, when massive ice sheets covered most of Canada and blocked southward migration. The genetic evidence suggested that the ancestral Native American population had been isolated in Beringia for as long as 8,000 years, from approximately 25,000 years ago when they diverged from their Asian source population to approximately 17,000 to 15,000 years ago when they began to move south as the ice sheets retreated. During that standstill, the population had diverged. The ancient Beringians split from the lineage that would become northern and southern Native Americans, music, and potentially other populations not yet sampled. Populations that may have gone extinct without leaving descendants also emerged during that long Beringian isolation. Music. The Trail Creek Caves remains were snapshots of that diversity. They were windows into a population structure, music, that had been completely invisible to archaeology before ancient DNA made it readable. The complexity continued to grow. In 2015, a study led by Pontus Skoglund and David Reich identified something even stranger in the genetic record. Some indigenous populations of South America, particularly groups in the Amazon basin, carried a small but statistically significant genetic component that appeared to derive from a population related to Australasians, the indigenous peoples of Australia, New Guinea, music, and the Andaman Islands. This component, called population Y, did not appear in most Native American groups. It appeared only in certain South American populations, music, and it had no obvious source. The Australasians had never been proposed as contributors to the peopling of the Americas. The distances were enormous. Music. The migration routes were unclear. The timing was problematic. Music. And yet the genetic signal was real. The most likely explanation was a ghost population, a group that had contributed ancestry to some early Americans, but had subsequently gone extinct without leaving any unmixed descendants. This ghost population might have arrived in the Americas separately from the main Native American lineage. It might have been absorbed into arriving Native American populations. It might have been displaced or eliminated by later arrivals. Whatever happened to them, their genetic legacy survived only as a trace, a faint Australasian signal detectable in a few South American groups and invisible everywhere else. The peopling of the Americas was not one migration. Music. It was not even two migrations. It was a process, a complex, multi-wave, multi-source process that unfolded over thousands of years, music, and involved populations that we are only now beginning to identify. The White Sands footprints pushed the timeline even further. In 2021, a team led by Matthew Bennett published evidence of human footprints preserved in the sediments of White Sands, New Mexico. The footprints were not artifacts that could be challenged on typological grounds. They were not animal bones that could be dismissed as naturally deposited. They were human footprints, the unmistakable impressions of human feet walking across a muddy lake shore. And they dated from approximately 21,000 to 23,000 years ago. The dating was controversial. It relied on radiocarbon dates from aquatic plant seeds, which can absorb old carbon from water and yield ages that are too old. In 2023, a follow-up study by Pagadi and colleagues confirmed the dates using multiple independent methods. The footprints were real. The dates were real. Humans had been present in the Americas at the height of the last glacial maximum, thousands of years before the ice-free corridor opened, thousands of years before the Beringian standstill was supposed to have ended, thousands of years before anyone was supposed to have been there. The implications were staggering. If the White Sands dates were accurate, humans had reached the interior of North America while massive ice sheets still covered most of Canada. The traditional ice-free corridor route was not available. The Kelp Highway, a proposed coastal route along the Pacific Northwest, might have been open, but the timing was at the extreme edge of what the coastal hypothesis predicted, and the genetic evidence did not clearly account for a population present that early. Were the White Sands people related to the ancient Beringians? Were they an even earlier unknown population that left no genetic descendants? Were they the source of the mysterious Australasian component? The questions multiplied faster than the answers. Jennifer Raff, the University of Kansas anthropologist and author of Origin, A Genetic History of the Americas, has synthesized the current state of knowledge. Her assessment is sobering. The simple stories are gone. The textbook diagrams with single arrows pointing from Siberia to the Americas are obsolete. Music. What remains is a research frontier, a field where new data arrives faster than consensus can form, where each ancient genome reveals both answers, music, and new questions, where the narrative of the peopling of the Americas is being rewritten in real time. The Trail Creek Caves remains are part of that rewriting. They are not unique. They are not even the most dramatic findings of the past decade, but they are representative. They show what Beringia looked like genetically at the end of the Pleistocene, a population structured into multiple lineages, some of which would become modern Native Americans, some of which would remain in Alaska, some of which would go extinct. They show that the founding of the Americas was not a moment, but a process, one that unfolded over thousands of years, involved multiple distinct populations, and left genetic signatures, music, that we are only now learning to read. The bones sitting in museum collections around the world, the remains that were excavated decades ago before ancient DNA was possible, contain information that has never been music extracted. The Trail Creek remains sat in a Danish museum for 60 years before being repatriated to Alaska. The genetic analysis became possible only after they returned. How many other collections contain similar data? How many skeletons excavated in the 19th and 20th centuries preserve genomes that could resolve outstanding questions about music, the peopling of the Americas? The answer is, we do not know. The ancient DNA revolution is ongoing. New techniques allow recovery from increasingly degraded samples. New analytical methods allow extraction of information from fragmentary genomes. New collaborations between geneticists and indigenous communities are establishing ethical frameworks for research on ancestral remains. The next decade will produce more data than the previous century, and that data will continue to complicate the simple stories. The Clovis first model is dead. The single migration model is dead. The ice-free corridor, music, as the exclusive route is dead. What remains is complexity. The recognition that the peopling of the Americas was as complicated as the peopling of any other continent. Modern research is pointing toward a much richer picture of the first settlement of the Americas than scientists once imagined. Instead of a single migration by one group of people, growing evidence suggests that several distinct populations reached the continent over an extended period, with each contributing to the genetic and cultural history of the Americas. Understanding that process has taken decades of research, and many questions are still being explored. The discoveries at Trail Creek Caves offer one important piece of that puzzle. They are not the only archaeological site changing our understanding of early human history, but they provide valuable evidence that complements discoveries made across Alaska and other parts of the Americas. Rather than relying solely on long-standing theories, researchers are increasingly turning to physical evidence preserved in ancient remains. With advances in DNA technology, those remains are now revealing information that was impossible to recover only a generation ago. The region surrounding Trail Creek Caves lies within the Bering Land Bridge National Preserve, an area recognized for its exceptional archaeological importance. During the last ice age, this landscape formed part of the land bridge that connected Asia and North America. As the climate warmed and sea levels rose, much of that ancient corridor disappeared beneath the waters of the Bering Strait. Countless campsites, hunting grounds, and other traces of early human activity were likely submerged, placing a significant portion of this history beyond the reach of traditional archaeological excavations. The same challenge applies to the proposed coastal migration route. If some of the earliest people traveled along the Pacific shoreline, many of the locations they occupied are now hidden beneath the ocean due to rising sea levels. As a result, researchers must rely on the limited number of surviving inland sites that escaped flooding. Locations such as Trail Creek Caves have therefore become exceptionally valuable because they preserve evidence from a period that is otherwise difficult to study. Scientific work at these sites continues today. Specialists in ancient DNA and archaeology, including researchers from several universities and research institutions, are constantly analyzing newly recovered samples. Every genome that is successfully sequenced adds another piece to the larger picture, sometimes confirming previous findings and sometimes raising entirely new questions about ancient migrations. What is becoming increasingly clear is that the settlement of the Americas was far more complex than earlier models suggested. Evidence now supports the existence of multiple migration events, diverse populations, and a timeline that stretches much deeper into the Ice Age than many researchers once believed. New discoveries, including ancient DNA studies and archaeological finds from numerous locations, continue to reshape this evolving narrative. Rather than replacing one simple explanation with another, scientists are assembling a far more detailed reconstruction based directly on physical evidence. Ancient bones, preserved DNA, and carefully excavated artifacts are gradually revealing how different populations moved across Beringia, entered North America, and eventually spread throughout 2 continents over thousands of years. Trail Creek Caves represents just one chapter in that enormous human journey. The caves remain a reminder that many of history's greatest discoveries are still waiting to be uncovered. Museum collections, archaeological archives, and research laboratories around the world continue to hold specimens that have yet to be fully analyzed. Each new genetic sequence has the potential to refine, expand, or even challenge our current understanding. This is why the story of the first Americans is still evolving. It is no longer driven primarily by assumptions or simplified historical models. Instead, it is being built from a growing body of archaeological discoveries, genetic evidence, and scientific analysis collected from dozens of sites across the Americas. As that evidence continues to accumulate, researchers are developing a more accurate picture of one of humanity's greatest migrations. The emerging history is undeniably more complex than the version once presented in textbooks. Yet that complexity is exactly what makes the story so fascinating. With every new excavation and every successfully decoded genome, scientists move one step closer to understanding how the Americas were truly settled. And the evidence continues to reveal a past that is far more remarkable than anyone once imagined.